I'm Kelly, and this is Marija. And to kick things off this morning, we're going to talk to you about some of the tools that we use when we're wearing our data science hats. First, we'll give you a brief background about ourselves, and then Marija is going to describe an experiment that we're both working on and talk about how she uses data science for molecular identification and annotation, as well as bringing together multiple types of omics data. Then I'll talk about a different part of that same experiment and show you one of my favorite tools for data visualization. So my background is in biostatistics, and much of my technical work here at PNNL has involved the development of methods, R code and R packages, as well as user interfaces as a front end to those R packages to help others use those tools without needing R programming. So now I will turn things over to Marija.
My name is Marija. It's green. Can you hear me now? Oh, okay. Good morning, everyone. My name is Marija. I work in EMSL as a chemist. I'm more on the sample prep side, but today I'm gonna talk about some tools that I use for data science for omics data. So omics data basically is information generated using science that ends in omics. If we want to basically profile the genome, then we are talking about genomics. If we are interested in RNA transcripts, then we are going to take transcriptomics approach. Or if we want to profile proteome or metabolome, then we are talking about proteomics or metabolomics approaches. So here in these slides are outlined some steps of experimental workflow that we use. So first step would be basically to write the experimental design and to plan our experiment. Then the second step would be to obtain the samples and to do the sample prep. So it all depends which omics approach we are going to be doing. After that, those samples we would run on the mass specs and acquired some data. And basically that's from where omics data fun part starts. Those acquired data sets are then compared against databases to do some molecular identification. If we want basically to compare the retention time on fragmentation to the database to really identify molecules, or if we are doing annotation, we are putatively doing putative association of the compounds to the molecular formula. Then next step would be to do some quality control and basically to keep the annotation and identification that we are confident in. And next step would be to present our data in the way we want, basically to summarize and visualize our data and to do some statistical analysis. As Kelly mentioned, I'm going to talk about a tool that I use for molecular identification and one tool that I use for annotation for one very interesting project. But before I start talking about the project and all of those tools, I would like to just briefly go over two approaches that are used in mass spectrometry-based omics analysis. So Kelly later, she's going to talk about omics data that was generated using bulk approach. And in my first part of the talk, I'm going to talk about data that was obtained using near single cell analysis. So if you imagine this fruit cup as a tissue sample, and if you homogenize it, you're gonna just get a smoothie, and you are basically average out all the molecular signatures. If you imagine that each fruit is a specific cell, for example. As you can see here in the image, if you're looking at the bulk sample, you are highly likely to get the bulk average. And most of the time, those small molecules that are lower abundant, they're likely be undetected. So the second approach that I use is basically to sort each of those fruits either as a separate fruit or in the fruit like groups, and then to independently analyze them. And like that, you are gonna keep basically the all molecular signatures that you're interested in. So there are some pros and cons for both. It all depends what are you looking for. But yeah, we are gonna show you data obtained by all approaches today. So let me tell you for a few words about these really interesting projects and basically why we leverage mass spectrometry capabilities we have here in EMSL to investigate the spatial distribution in one very complex and heterogeneous system such as leafcutter and fungal garden ecosystem. So leafcutter and fungal garden ecosystem is naturally a whole microbial ecosystem. And I'm quite sure that most of you have seen those tiny ants carrying a leaf across the rainforest floors. What is really interesting for those tiny ants is that they are little farmers. They do cut and collect the leaves and store them in the underground structures called fungus garden. But they do not use the leaves as like food. Instead, they grow the fungus in the garden, and basically fungus break down lignin to small aromatic compounds and also hemicellulose and cellulose to high energy sugars. Those compounds are then further converted by resident bacteria that are also in the garden to produce some high value bioproducts. So we were really interested in our research to look at the spatial organization, and we wanted to see basically how the spatial organization looks within the garden. And we also wanted to basically profile molecules in some specific snapshot of the time and to see what are the molecular mechanisms that are behind the lignocellulose degradation. One really interesting thing about this fungus garden is that there is a vertical gradient of lignocellulose degradation within the garden, which means that at the top of the garden and at the bottom of the garden, basically, you will find similar protein, but they won't be active. Like it won't be active, the same enzyme at the bottom and at the top and at the very bottom, because of that vertical gradient, which means that if we use a single omics technique to profile one specific part of the garden, we are gonna definitely detect a lot of enzymes there, but we really want to know which enzymes were active at that specific moment. That's why we use another spatial technique to profile the metabolome, and we basically came up with a workflow that is called metabolome-informed proteome imaging workflow, where we use two complementary spatial techniques to profile metabolome and proteome in order to really see what is going on at that specific point of the time at the spatial level in that tiny part of the garden. So we took a little part of fungal garden, we embedded it and cryosectioned it. So one section was used to profile metabolome, basically to spatially resolve metabolites by using multi-mass spectrometry imaging techniques. After that, we leverage basically the platform called Metaspace to annotate each of those metabolites and to select and to try to find lignocellulose degradation hotspots. After we identify some lignocellulose degradation hotspots, we use the adjacent section to profile each of those hotspots regions by spatial proteomics and to see which enzymes are there. For that, we use our spatial proteomics workflow. After that, we use metaproteomics analysis basically to do identification of each of those peptides that were profiled in those regions and to do functional and taxa assignment. And in the end, we integrated our spatial metabolomics and spatial proteomics analysis and to reveal some underlying biological pathways. And basically today I'm going to talk about molecular annotation tool, then metaproteomics analysis and our integration, because this is more data science. So for molecular annotation of mass spectrometry imaging data, we use Metaspace, that is open source framework that is based in 3 principles. The first one is to compare basically our mass spectrometry imaging, multi-mass spectrometry imaging data against the database. And to do the annotation. Here I really have to clearly say that multi-mass spectrometry imaging data is MS1 based, so we don't have fragmentation data. And we, in EMSL, we have some high mass resolving power mass spectrometers that we use. And from there, we can really have we can measure accurate molecular mass, and from there we can really ascribe accurate molecular formula. But there are still some mass isomers that we cannot distinguish, so that's why we are here talking only about molecular annotation. So the second step would be for each of those annotation to score, and basically to check how close the theoretical properties match the detected ones, and to check the quality of the image. So if you could see here, this is an optical image of one fungal garden cryosection. And here they're overlaid optical image within ion images obtained by multi-mass spectrometry imaging. So for MSM, molecule signal match score, it's basically compute and check how well theoretical and experimental properties are matched for each of isotopic pattern. So basically check how well it correlates the spatial image with each of those isotopic peaks, and also it looks how well the relative abundance of each of those isotopic peaks match between experimental and theoretical. Next thing is to do the estimation of ratio of false positive and in results to basically do the FDR calculation. And after that, we can just do basically some kind of QC to set up the threshold, so we can keep the ones that has FDR that are lower than 10%. So this is how the metaspace interface looks like. For our specific project, we use KEGG database, but if you're doing metabolomics, you can use Natural Product Atlas or some other databases. So if you are interested in lipidomics, you can use Lipid Swiss, or you can like curate your database and use like custom database. As I previously mentioned, we can overlay ion image with optical image, and this is how basically multi-mass spectrometry ion image looks like, and we overlay it with the optical image so we can see each feature there. As I previously mentioned, we are here talking about only annotations, and you can see here for each of those M/Z values, molecular formula that is ascribed, and all the combination of annotated molecules. You can also see here their structural formulas. As I said, those are mass isomers. We could not distinguish them by doing only MS1. There is also MSMS score for each of those. And if you go in diagnostic, you can see how it's been calculated, and also FDR, we set it up at 10%. Our multi-mass spectrometry imaging data map the presence of low molecular weight products. And basically all low molecular weight lignin products that were detected, they had a spatial pattern that is here presented on the left-hand side ion image. And from there, we mapped some, we found some lignocellulose degradation hotspots, and we named them lignin microhabitat 1, 2, and 3. And we wanted to further profile them by doing spatial proteomics. There is also one spatial pattern that was really widely observed. It was really abundant in our data. And that spatial pattern was only localized in one specific feature. And basically all metabolites that were annotated to that one were primary metabolites. And by looking at the looking under the microscope, that feature really looked to us that could come from ant, because it looks like ant wing or ant egg. So we also wanted to profile that one by spatial proteomics, just to see what it is. Then we follow our spatial proteomics workflow. Basically, we cut each of those 4 regions of interest, and we did replicates and prepare them using spatial proteomics workflow, and acquired some peptide data sets by using mass spectrometry analyzers. For For peptide identification, we used metaproteomics approach. Metaproteomics is an umbrella term for all the experimental approaches that study microbials, communities, and microbes that are coming from environmental sources. In our specific cases, we curated our reference database that contained more than 50 million proteins of known members of this fungus garden consortium. So we downloaded FASTA files from JGI website. After that, we clustered those proteins into more than 24 million clusters in order to get the database, to reduce the size of the database. Then those cluster proteins were annotated again using KEGG database. So we wanted to do taxa and functional annotation on those. Then previously acquired mass spectrometry peptide data was compared against our custom database using MSGF plus search engine. And that search engine, basically, it first matched the MS1 that is within 20 or whichever instrument you use, but in our case, we use within 20 ppm. And basically for all MS1 matches that match with our reference database, we then compare our fragmentation pattern and provided, and we obtained basically probability score for each peptide spectrum that were matched with our database. After that, we did some further filtering and kept the only peptide that had FDR percent less than 1%. Then we did another kind of filtering through when we kept the peptides that are unique and that basically for each of the cluster, it had to contain 2 unique peptides, and also each peptide had to be detected in at least 2 replicates. So that was our further criteria to filter out some results. In the end, our metaproteomics data reveal really a complex community. And as you can see here in lignin microhabitats 1, 2, and 3, the most abundant were enzymes that were coming from fungus. And by looking here at this table, you can see that the purple one were high abundant, and those are enzymes that were involved in carbohydrate metabolism as it was expected, because from the previous experiments, we know that fungus is basically the main driver of lignocellulose degradation. What was really interesting that for that primary metabolite microhabitat, as we called it, we really so that was the feature that were coming from ant, and by doing some deeper literature research, we realized that that was basically ant egg. In the end, we integrated our spatial metabolomics and spatial proteomics results to reveal some underlying biological pathways that were occurring in those tiny microscale regions. So we mapped the detected metabolites and the paired enzymes in the respective KEGG metabolic pathways. From our Metaspace platform, we exported all the annotated metabolites, and those annotated metabolites, they had their KEGG compound number. On the other hand, from metaproteomics analysis, we did functional annotation, and we had the KEGG orthology number for each of those clusters. So it was really easy to put together and to integrate our spatial proteomics and metabolomics results. As you could see here, this is aromatic compound degradation pathway where we detected substrate, enzyme, and product, and they all really correlate really nicely on the spatial level. This is just the orthology number, KEGG orthology number. You can see here all the brightened pathways that KEGG functional ortholog was involved. The other pathway, just as example, is ring cleavage pathways, where we detected two substrate and 2 different enzymes that are converting those substrate to the same enzyme, to the same product, to malyacetate, as you could see here in those ion images, both substrate, both enzymes, and the product, they correlate really nicely. So they're detected in all three green regions that are microhabitat, and they're not detected in the white region, which is ant region. And that is basically my first part of the talk. Kelly now, she's going to talk about bulk omics data. Thank you.
Thank you. Right. So as Marija said at the very beginning of her talk, she does the instrument side and the molecular identification annotation. Then there's some quality control filtering, data normalization that occur. And then this is typically where myself or another statistician or data scientist would take over to do additional visualizations and statistical analyses. So I'm going to talk to you about how we use visualization sometimes to help inform the analyses that we do. So Marija was doing her near single cell work on this native fungus. And there was also really a great interest in understanding how this system responds when it's under stress. So to induce stress, what they did was introduce a pathogenic fungus to infect this fungal garden. And this photo shows a sample from one fungal garden where a healthy control sample was taken prior to infection from this left-hand side. And then on the right-hand side of this tube, a pathogenic fungus was added, and it was allowed to spread across the tube until it reached the midway point. And that midway point was determined by visual expert inspection. And at that point, the sample was frozen to stop the infection from spreading further. Once the sample had been frozen, individual subsamples were taken across that infection gradient. And in this system, a bulk analysis approach was used, so that fruit smoothie, because it's just too complex and too large of a sample for it to be realistic to image all parts of this tube. So that's one advantage of using a bulk over a near single cell or a single cell approach. From these samples, the aim was to determine how the individual biomolecules, so for us, lipids and proteins, changed across the infection gradient. So to do this, we needed to know how to model those individual biomolecule abundances across the gradient. We could imagine that there might be different patterns, right, for different molecules. Some lipids might show an increase in abundance across that gradient. For others, there might be no change, or we might have some nonlinear relationships as well. The challenge is that we have hundreds or thousands of biomolecules in our data set. So if we were to make individual plots for each lipid or each protein and try to flip through and sort of mentally catalog the different patterns, it would take a long time and it would probably be very overwhelming. So this is where a tool called Trelliscope comes into play. And what this tool does is it provides a trellis display. And what that means basically is that you have one graph for every unit or every lipid in our case, and each graph is identically structured. So as soon as you understand what one graph is showing you, you'll understand what all of the graphs are showing you. And this type of configuration can really help us spot similarities, differences, surprising things. And there's a famous example of where a trellis display revealed something unexpected or previously unknown, and that is in the Fisher barley data. So that's what you're seeing here on the screen. And this data set contained the average barley yield for 2 different years at 6 different sites. So here we have one plot or panel for each of those 6 sites for 10 different varieties of barley. So the y-axis on these plots show the different barley varieties, and the x-axis shows you the yield. The color denotes the year. Originally, someone had graphed this information by hand, probably on separate pages in a lab notebook, but it wasn't until making a trellis display where you could see all of these graphs side by side 60 years later that this circled graph was sort of noticed. And what they realized is that there had been an error when they were recording data for this particular site, and the years had been swapped accidentally. So this just goes to show that how you view your data can be really important, and it can have a big impact on your understanding of it, and it can help you catch errors that sometimes occur in experiments. So Trelliscope leverages these types of trellis displays, and more than just seeing a bunch of plots sort of all at once, it lets you connect information about the samples, information about the biomolecules to each plot so that you can sort and filter through your plots and really start to understand what's going on in the data that you're looking at. It also provides a really nice way to share this display with collaborators so that other people can start to really dive into the data as well. Okay, so now I'm going to show you an example of a Trelliscope display that we made for this project that Marija described, and specifically for the bulk omics portion. Live demo here. Okay. So right now I have pulled up a Trelliscope display that we created, and we're seeing a graph for a single lipid. And there are 6 different fungal gardens represented here by the different colors. The y-axis shows us the log2 normalized abundance for this particular lipid. And the x-axis is representing that infection gradient. So a low infection on the left-hand side, high infection where that pathogenic fungus was introduced on the right. And this vertical line at 0 represents that midpoint where the sample was frozen. So you might notice some of the lines start sooner or end later than others, and that's because that midpoint wasn't identical for each of the fungal gardens. After sample collection and some analysis, they were able to determine precisely where that midpoint was, so we've aligned each of the fungal gardens on this graph. So I can now leverage Trelliscope to show more than one plot at a time. Up here, there we go. And right now we can see that there are 766 lipids in this data set. So each of these plots is for a different lipid. We have the ability to add different information to the bottom of the plots. Right now we just have lipid name, but if we wanted to include lipid class, we could do that. And it's possible to define your own metrics and information that you can include so that it really suits the type of experiment and data that you're looking at. So right now, we are looking at all of these plots sorted by lipid name, just alphabetically. But it might be that we are really interested in sorting by something else. So one of the things that we did was compute a log2 fold change from that highest infection point back to that control, the healthy fungal sample. And we've included that here, so we could change what we're sorting on. So log2 fold change of that high infection versus control. We're going to remove our lipid name. So right now we're sorting from lowest log2 fold change to highest. And we can sort of page through and look at different patterns that we see. We can also filter. So if we're only interested in looking at a single lipid class, for instance, at a time, we can filter down just to the triglycerides, say. And now we have 88 lipids that we're currently viewing. So again, we can page through, we can see some of these look like there's a slight increase. Some of them look more flat, like there's not much of a change across that gradient. We remove our filter and sort in the opposite direction. We start to see some very different looking patterns in our lipids. So this gives us a really quick way to sort of take in the whole data set all at once and informs the types of models that we might need to be using in order to model that infection gradient. Now I will go back to this. So if you're interested in making one of these displays for your own work, there are quite a few different options available to you. So the most flexible option is to program directly in R using the Trelliscope JS package. This, while giving you the most flexibility, also requires the most effort and expertise in order to build. You can also go through quality control normalization workflow in the PMART-R package. And that package includes a function to generate a predefined display with just a single line of code. So that would be another option available. And there are also two different user interfaces that are available online. And I think you're going to hear a little bit more about these later today and then also throughout the week. With that, we'd like to acknowledge all of the project team and Dr. Kristen Burnham-Johnson, the PI, our collaborators at the University of Wisconsin-Madison, and the funding source. We're happy to take any questions that you have.
Hi, I have two questions. You mentioned during the proteomics workflow, creating a reference database with more than 50 million proteins, downloading FASTA files from JGI. Did you use like KBase for any of this? Because I'm just trying to understand like the programs that you did for that workflow.
So that was all our computational scientist. So she basically did all the work. She downloaded all those FASTA files and clustered them together.
Awesome. And then on the last presentation, you mentioned two user interfaces, mode and map. And then you had this really cool display with all of the graphs and filtering and everything. Was that either mode or map?
The display that I showed you was one that we created from within R ourselves.
Okay, cool.
But if you go through either of those user interfaces, you'll get something that looks very similar. Yeah. Same sort of interface.
Thanks.
Mm-hmm.
Are there any questions online?
Is someone monitoring that though for us and can read them out? Okay, great. Thanks.
Okay, no more questions then.
All right, thank you.